Hello everyone, welcome to my YouTube channel Pharmacy. Today we will start our new topic, that is transport of molecules across cell membrane. Lecture will include Introduction to transport across cell membrane, passive transport, types of passive transport, active transport and types of active transport. So, let's start. Contents of cells are completely surrounded by cell membrane or plasma membrane. So, communication between cell and external environment is essential. And transport of molecules or ions across plasma membrane occurs. Functions of plasma membrane are as follows. It retains the dissolved materials of cells to prevent leaking, and exchange of materials into and out of cell also takes place. This slide shows the simplified classification of transport across cell membrane. It is of two types, passive transport and active transport. Passive transport is further classified as simple diffusion, facilitated diffusion and osmosis. In simple diffusion, transport of molecules or ions from higher concentration to lower concentration without consumption of energy. Next is facilitated diffusion. In this, transport of molecules or ions from higher concentration to lower concentration through specific protein channels and no ATP is consumed. Last type is osmosis, transport of molecules or ions from higher concentration to lower concentration through a semi-permeable membrane, and the solvent used is water. Next is primary active transport. In this, single molecule is transported at once, and other type is secondary active transport. In this, two types of molecules are transported at once and co-transporters are uniport, synport and antiport. Now, we will understand this classification in detail. So, first one is passive transport, movement of molecules or ions from higher concentration to lower concentration. It transports water-soluble molecules such as oxygen, carbon dioxide and lipids. It is generally bidirectional and no expenditure of ATP. It is a slow and spontaneous process. First type of passive transport is simple diffusion. It is similar to passive transport. In this, transport of molecules is from higher concentration to lower concentration without ATP consumption. It is a spontaneous process and it maintains the turgidity of cell. Second type is facilitated diffusion. It is defined as the transport of molecules from higher concentration to lower concentration without expenditure of ATP. Transfer occurs through specific protein channels. Factors affecting facilitated diffusion are temperature. As the temperature increases, the movement of molecules increases due to an increase in energy. Second is concentration. The movement of molecules takes place from the region of higher to lower concentration. Third is diffusion distance. Diffusion rate is faster through smaller distance. Last one is size of molecules. Small molecules are lighter and hence diffuse faster than larger molecules. Examples are aquaporins and glucose transporter. Third type of passive transport is osmosis. Transport of molecules from higher concentration to lower concentration without expenditure of ATP through a semi-permeable membrane, and in this, solvent used is water. It is a slow and spontaneous process. Transfer of molecules or ions is in the direction that it tends to equalize the solute concentration on both sides. There are two types osmosis, that is endosmosis and exosmosis. In endosmosis, when a substance is placed in a hypotonic solution, the solvent moves inside the cell and the cell becomes turgid. In exosmosis, When a substance is placed in a hypertonic solution, the solvent molecules move outside the cell and the cell becomes flaccid. And there are three types of solutions, that is isotonic, hypertonic and hypotonic. In isotonic solutions, there is same concentration of solute inside and outside the cell. In hypertonic solutions, there is higher solute concentration outside the cell than inside. In hypotonic solutions, there is higher solute concentration inside the cell than outside. This diagram shows the tonicity of red blood cells and see what happens if RBCs are placed into hypotonic, isotonic and hypertonic solution. In figure A, hypotonic solution has lower solute concentration compared to the intracellular solute concentration. When RBC is placed into hypotonic solution, there will be a net movement of free water into the cell. This situation will result in the increased intracellular volume with a lower intracellular solute concentration. The solution will end up with a higher overall solute concentration. In figure B, solution has same solute concentration compared to the intracellular solute concentration. When RBC is placed in an isotonic solution, there will be a no net movement of water towards the solution or the cell. In figure C, solution has higher solute concentration compared to the intracellular solute concentration. When RBC is placed in any hypertonic solution, there will be a movement of free water out of the cell and into the solution. After the solutions are allowed to equilibrate, the result will be a cell with a lower overall volume. The remaining volume inside the cell will have a higher solute concentration and the cell will appear shriveled under the microscope. Now, our next topic is active transport. It is defined as the transport of molecules or ions against concentration gradient, that is from lower to higher concentration. It requires energy as ATP and essential for various physiological such as nutrient uptake, hormone secretion and nerve impulse transmission. Active transport is highly selective and regulated with different transporters specific to different molecules or ions. Examples of active transport are sodium-potassium pump, movement of calcium ions out of cardiac muscle cells, transportation of amino acid across the intestinal lining in human gut, WBCs defend invading diseases and phagocytosis of bacteria by macrophages. This diagram explains the mechanism of active transport. Materials require a specific protein that acts as a carrier molecule for the substrate to pass through membrane. On one side of surface, the carrier represents its binding site. On binding with the substrate, the carrier undergoes a conformational change. This transports the carrier-bound molecule to other site of membrane where it is discharged in an unaltered state. The carrier is left behind the membrane. This process requires energy, which is supplied by ATP. In this way, active transport enables the transport through the membrane. Active transport is of two types, that is primary active transport and secondary active transport. In primary active transport, transport of molecules or ions is against gradient, that means from lower concentration to higher concentration. It is also known as direct active transport. It directly uses chemical energy as ATP for transportation. A single molecule is transported at once. Transmembrane proteins are unique to the ion transported through them and includes ion pumps and ion channels. Energy used in this process are ATP, redox energy, and photon energy. Examples are sodium-potassium pump, calcium pump in muscles and proton pump in stomach. Biological importance of active transport is to maintain the right concentration of ions or molecules in living cells. Now, our last topic is secondary active transport. It is defined as a form of active transport in which a transporter protein couples the movement of ion. It is also known as coupled transport or co-transport. Transport of two different molecules at a time takes place and energy is not directly used for movement. Transport is powered by this energy from electrochemical potential difference as ions are pumped into and out of the cell and no direct coupling of ATP. One ion is allowed to move down its electrochemical gradient. This results in increased entropy that can be used as a source of energy, and depending on the direction of movement of two substances, cotransporter may be uniport, symport and antiport. Examples of secondary active transport is sodium ions moving down the electrochemical gradient across the plasma membrane power up the transport of a second ion against its gradient. It is used to store high-energy hydrogen ions for ATP production. In next lecture, we will understand the concepts of cell division. Thank you. Keep watching and take care.